So I am going through tissue culture. In this process, a very small part should be collected from the buds, shoots, shoot apex, or any other part of plant, which is called explant. And this is the propagation of plants involving cell and tissue culture. That is why it is called tissue culture technique or micropropagation. Another name of tissue culture is micropropagation. The portion of plants which is collected from any parts of plant is called explant. Now the explants are treated with sterilization chemicals to prevent microbial growth and then cultured in a particular nutrient medium. Then in nutrient medium, the cells grow and divide to form a mass of cell, which is called callus. And then some growth regulators, that is plant hormones, are to be added with the callus. Then the callus differentiates into plant parts looking like a tiny plant, which is called plantillet. Once again I am repeating, the cells grow and divide to form a mass of cell, which is called callus. Then with the callus, some growth regulating hormones are to be added. Then obviously the callus differentiates into plant parts looking like a tiny plants. These are called plantillets. Now after 6 to 4 weeks, the plantillets can be transferred from the culture medium to the soil. So this is tissue culture, and the method is called tissue culture technique. It is being used in orchids, gladiolus, potato, sugarcane, etc. These are the examples. Now we have completed already vegetative propagation. Some few points are given here regarding the advantages of vegetative propagation and disadvantages of vegetative propagation. At first, advantages of vegetative propagation. Vegetative propagation, number one, I am going through the points given here. It is a cheaper, rapid and convenient method of growing plants. I think you can understand the meaning of this point. Then 2 number, it is useful for production of seedless plants such as sugarcane, grapes, pineapples. That means the plants which do not produce seeds, they can be propagated by using vegetative propagation. Number 3, desirable characters of fruits and flowers can be maintained in new plants by vegetative propagation. That means if you think that the parent plant having the characteristics, having the desirable characteristics, it can be restored in the offsprings. That is why the new plants that we are getting from the parent plant by this method, the new plants contain all the characteristics of their parent plants. Then disadvantages of vegetative propagation. It does not produce new varieties, though the plants are produced from single plant, only one plant. That means there is no chance of obtaining any variations or new varieties. So it does not produce new varieties. The offsprings are same as their parent plants. Number 2, it leads to overcrowding around the plant, leading to less light and food. That means if a lot of plants are grown around the parent plant, then they will not get proper amount of sunlight and proper amount of food for their growth and development. So this is also a kind of disadvantages of this kind of propagation. Then number 3, plants produce are genetically alike, hence they are susceptible to the same diseases. That means if the parent plants are having any disease, the same disease will be in the offsprings, I mean new plants. We cannot avoid it. It is also a kind of disadvantage. So number 3 is completed. Number 4, undesirable characters are transmitted from one generation to another. If the parent plants are having undesirable characteristics, these undesirable characteristics will be transmitted into their offsprings. So these are some of the disadvantages of vegetative propagation. Now I have completed asexual reproduction in plants. Next day I will discuss about the sexual reproduction of plants. But dear students, you have to go through the book very nicely, read each and every line. I mean, you should read the chapter thoroughly and try to understand all types of asexual reproduction with example, and practice some diagrams from this portion like budding, fission, then fragmentation. All diagrams you just draw in your class or copy. And now you are having a lot of time, you are getting ample time, so you can try to grow some plants at your home, for very common plants which are available at your home, by the method of vegetative propagation. For example, you can use the method of cutting. You can use tuber stem, tuber root to grow some plants. You can grow some plants by using layering method. So tissue culture is not possible, grafting is little difficult for you. So some methods can easily be used by you at home and grow some plants. I hope you will grow some plants by using all kinds of at least some kinds of vegetative propagation or asexual mode of reproduction.